So hello, my name is Nadav, and this is Sanketh. And today we're going to present to you experiment-guided generative models for protein structures and dynamics. So in the last few years, there's been a lot of progress in the field of protein structure generative models, specifically AlphaFold, as most of you probably heard about. And in this work, we use AlphaFold 3 to solve some cool inverse problems in structural biology. So let's first start with the baseline. So what are proteins? So proteins are the building blocks of life, the little machinery, and they're comprised of amino acids, which are like Lego blocks for the protein. So you use these Lego blocks, these amino acids, to build some 3D shapes, and these 3D shapes are the proteins. So in this image, we see some simulation of a cell, and all the little pieces are proteins. So proteins are responsible for all the interactions and actions in a cell, and the protein 3D shape controls the functions and dynamics. Protein. So how are proteins created? So the first stage is called transcription, where the DNA splits and RNA nucleotides fits the split region and create a chain called the mRNA. The M stands for messenger. The mRNA travels to the ribosome where translation occurs. So each three letters in the mRNA is called a codon, matches an amino acid. So a chain of amino acid is created by fitting these little molecules called the tRNA, which have a codon that fits the codons in the mRNA and an amino acid. After the chain is created, the chain falls into a 3D shape, and that is our final protein. So in this animation, we can see 4 times sped up, a simulation of a peptide chain going out of the ribosome and folding. So yes. So what we can understand from this, that the sequence of DNA or the sequence of amino acid, is the one that determines the 3D structure of the protein and hence affecting its function and dynamics. So a common technique for determining the 3D structure of proteins is called X-ray crystallography. So in this technique, a sample of the protein is purified and then the protein is crystallized. The crystal has a repeating pattern of a repeating cell, which is called the unit cell, and it's repeating according to some symmetry group. So X-rays are emitted on the crystal and diffract, creating some diffraction patterns. The diffraction patterns are used to determine the norm of the structure factors. So what are structure factors? Structure factors are the Fourier coefficients of the electron density of the content of the unit cell. The phases are added using some different techniques, and the 3D electron density is reconstructed. We will denote this reconstructed electron density as FO, O as in observed. After FO is calculated, an atomic model is fitted manually to the density cloud, and this is the protein structure that is deposited to the PDP, which is the protein data bank. So how do we get from an atomic model to an approximate electron density? So we do this by a mixture of 5 Gaussians around each atom. The mean, the variance, and the scale depend on the atomic element and the B factor, which simulates the movements of the local movements of that atom. And an atomic model fits the electron density when FO and FC are close. This representation of proteins represent the protein as a mixture of marginal atom distributions. So each atom is represented by a center and a B factor. The joint atom distribution is not modeled as it is very complicated, but if we would sample from the atom distribution of atoms while neglecting the joint distribution, meaning no covariance, we would get non-physical protein structures. This is important because a standard assumption is a single crystal has a single protein conformation. But as we can see in this MD simulation of the SARS-CoV spike proteins, the protein vibrates and changes its conformation a lot. So when we observe the electron density of proteins in a crystal, we observe the average electron density of an ensemble of conformations of the same protein. In this image on the right, we can see that the center of the density is an atom, and the density around it represents an ensemble average density across the different conformations in the crystal.
So another modality to sense, so X-ray crystallography measures how a protein structure is. So another modality that is common in in structural biology to sense proteins, let's say, is NMR spectroscopy. So the NMR spectroscopy exploits the property of nuclear magnetic resonance. This is the physical principle it exploits to sense the nuclear spin. Nuclear spin is an intrinsic quantum property of an atom. So each nucleus can be thought of as a little magnet and whose magnetic field is influenced by the local magnetic environment. So when these nuclei are placed in a strong magnetic field, the nuclei either sorry, a strong magnetic field, the nuclei either align with or against the magnetic field. Then once these nuclei are aligned, then a radio frequency pulse is applied to excite these nuclei. Um, this the nuclei that are resonating with the frequency of the radio frequency pulse that is imposed, uh, they absorb energy and they emit it once the pulse is induced. Just a moment. Yeah. So once the nuclei return back to the equilibrium, they release an electromagnetic radiation, which is equal to the frequency of their resonant frequency. So these frequencies are sensed essentially, and they're called chemical shifts in NMR, and the intensity corresponding to the electromagnetic wave which is released is is called the is dependent on the number of atoms that are releasing the same chemical shift. So because proteins are large molecules, the electronic environment and the electronic environment around each nuclei is different. Every atom exhibits a different and distinct chemical shift. For example, in this case, if you take a small let's say tripeptide, a carbon has a different chemical shift than a C alpha that compared to a carbon that is present in a side chain. So, and they're all measured in parts per million. So the difference is very subtle and yet distinguishable. So the key thing in NMR is you observe these peaks, but you don't know which atom they're corresponding to. You have your sample in a solution. You're measuring these peaks at different frequencies, but you don't know which atom corresponds to which peak. So the key challenge in NMR is to assign the chemical shifts of different atoms in the molecule to different spectra that they emit. So this is usually the most time-consuming and challenging part of an experimental protocol in NMR. And the key idea behind how this thing is done, because given that it's complicated, is by exploiting the phenomena of magnetization transfer. So when an ARF pulse is applied and a particular nucleus absorbs its resonant the energy, then it also, while relaxing, this nucleus while relaxing, it also transfers the magnetization to neighboring atoms. For example, those that are bonded that are linked through a covalent bond. So once you induce an RF pulse, a certain atom absorbs the radiation. Then you start screening all the atoms which at all the frequencies at which you receive this electromagnetic wave back. Then you sense probably that a linked atom through covalent bond also emits an RF an energy back at its own resonant frequency. For example, this can be denoted as a cross peak. Essentially, the fact that you see a red dot here means that you co-sensed the fact that this carbon, when it has a chemical shift of a certain ppm, this nitrogen also emitted a chemical shift of different ppm. So this procedure, then later, when a nitrogen is excited, it can sense that another carbon is attached to it with a different chemical shift. So by this, by acquiring these two measurements, you know that this carbon is attached to this carbon via through nitrogen. So essentially, you're kind of stitching together like the which frequencies are attached to which which frequencies are attached to which atoms. And this is crucial before you do anything with this data. Um, so another way so not only does the magnetization transfer happen through covalent bonds, but it also happens through space. This is called the nuclear Overhauser effect. It's a phenomenon in NMR where the intensity of a nuclear spin changes based on another spin, which is if there is another spin in the environment that is excited. So they're called dipolar couplings, which means that two magnets are coupled because they are spatially in spatial proximity, and this coupling can be sensed in the same way as before. So the intensity, if you have two hydrogens for which you observe a cross peak, it means that they are linked in space via the intensity indicates how close they are in space, and this intensity decays by the sixth power of the distance of their proximity. So effectively, anything beyond 5 angstrom radius you can't sense it. But you can sense interactions of two nuclei which are 5 angstrom apart. So another crucial detail here is that the intensity of the cross peak that you measure here is a function of how this protein moves. It's a temporal average of the distance between these two atoms across time. So in some sense, just as electron density, it's not a single molecule measurement, it's an ensemble measurement. So in the same way, the cross peak intensity is an ensemble measurement, and which is averaged across time. So so in some sense, what we want to say is that most of the structural structural biology measurements, like both electron in not cryo, but X-ray crystallography and NMR, are ensemble measurements. So you need to really natively treat this fact. So
below the 5 angstrom threshold, is this distance average really well resolved, or is this now almost also still like a contact rather than a distance?
it's inferred from the intensity. So when you observe a peak between two hydrogens, you
Yeah,
observe
sure. But how confident are you then in that being the average distance? Like, how can we be like, aha, within 0.1 angstrom, I'm sure this is actually the mean distance? Or is it more like there's this experimental error and that experimental error?
So usually these intensities are decoded into bounds, like upper and lower bound. You know for sure that these both atoms are linked by, if the assignment is done correctly, you know for sure that the distance between these two atoms in an ensemble is between this upper bound and this lower bound.
Okay.
So yeah, it is but it is all subject to the fact that the assignment is done correctly, right? If the assignment is not true, like in the sense that somebody assigned this peak wrong, then you have a wrong measurement. So if the assignment is true, then you know how at least where this distance lies.
Yeah.
Thank you. All right, so there has been a groundbreaking result in the field about a decade ago. You probably heard about it because it won a Nobel Prize, and it led to the development of AlphaFold. So this result allows for the creation of accurate sequence-to-structure protein models. So the task at hand is predicting the protein 3D structure purely from the protein sequence or the sequence of amino acids. So specific amino acid sequences will appear in different organisms with small variations due to evolution. If we were to create a database of similar sequences and align them together, we would notice that there are pairs of indices that mutate in correlated manner. The hypothesis is that evolution co-evolved these residues to preserve a specific structure or function because they have some magnetic interaction or they are in contact. Second. These coevolution maps can be used to predict a spatial contact map from this evolutionary data. So how does AlphaFold 3 works? So you got a sequence going in for multiple sequence alignment search or MSA search. The result of the search is inserted to a model called the performer, which embeds this, and the embedding of the performer is used in the diffusion model as conditioning. The diffusion model then uses a standard diffusion process to get the 3D protein structure. But there are a few problems with this approach. So first of all, AlphaFold 3 was trained partially on old data that could have been poorly fitted or could have been acquired using old techniques or equipment which don't give the best measurements. And also, AlphaFold 3 was trained under the assumption that a single sequence yield a single structure. So we are introducing experiment-guided AlphaFold that solves the inverse problems from experimental observations. So what's the difference between our approach and the standard approach? So the standard approach models the proteins as a single conformation where each atom has a variance of its position. But our approach models the density as the average density of a protein ensemble. This way, we sample from the joint distribution of atoms and not model each atom distribution separately. So how can we approximate the posterior likelihood? So using simple probability laws, we can split the posterior into two terms, the likelihood of matching the experimental results and the prior over the protein structures. The likelihood over the entire ensemble is interpretable because, as we've discussed before, our observations are over an ensemble of structures and not single structures, but the prior is over each structure separately, so we can calculate this prior in a separate manner. So in the case of X-ray crystallography, we can approximate the likelihood as the norm between the observed electron density and the average calculated electron density of our ensemble.
So in NMR, given the NOE measurements that we say, the spatial dipolar couplings, if you were to determine an ensemble, the goal of a likelihood is to say, I observed this experimental measurement, which are the NOE couplings implied by the intensities, and I now generate me an ensemble which produces these intensities. So typically how it is done is, as I explained earlier, so you convert these intensities into upper and lower bounds on the distance of the pairwise atoms post-assignment. And then it is enforced that the average distance within the ensemble should fall within these bounds. If it does fall within these bounds, it is adhering to the experimental measurement, and it doesn't incur any violation. So if not, we impose some error based on how far it falls away from the experimentally determined structure, experimentally determined constraints. So essentially, the problem of NMR structure determination is that given all the NMR observables, you want to determine what ensemble produced them. So yeah, essentially that's the inverse problem.
So for our prior, as we discussed before, we are going to use AlphaFold 3, and we're going to use the diffusion model inside. Of course, we're going to use the performer and the MSA embeddings, but that is transparent to us. So we're going to use a standard reverse diffusion process. So we start with pure noise and integrate the diffusion SD to retrieve a noiseless protein sample. In this SD, the most important term is the score function that derives the flow or the drift. So how do we put everything together? We will start by expanding the current diffusion SDE over multiple protein conformations. This is very simple using basically matrix notations. But we introduce the experimental likelihood function as a guidance term in the SD. We integrate this SDE and we will render protein structures that are extremely faithful to the experimental observations. So in summary, how does our entire pipeline look like? So it starts with an amino acid sequence, and using AlphaFold, we can compute an ensemble of proteins. We can use the proposed forward models to compute the likelihood term over the samples and use the derivatives of these forward models to guide the diffusion process. We add the force field relaxation or amber force field relaxation to correct slight bond length and angles as a post-processing stage. And we also add the use of a matching pursuit filtering scheme to find a small subset of the ensemble which fits best to the data. So we can think about it as biology goes here. We use some biological data to use the MSO, which is the multiple sequence alignment. And then we use the physical part, which are the measurements in this part. This is for the guidance. And then we use some prior from chemistry, also inside the diffusion model and also inside the amber force field relaxation. So let's go over some results. So here we can see COVID-19's 8th opening reading frame, which is notoriously the best reading frame. Yes.
Let's go back for a little bit.
Yes.
So what we want to do is to take your, or like what sort of experimental inputs are we talking about here? Are we talking about a very concrete, like many distances, well-resolved NMR distance matrix or just like a single distance that you observed? What does this look like?
So yeah, so in the case of, so we have both NMR and crystallography. So in the case of NMR, we receive distance restraints, which are decoded from densities, right? And we have a list of them. You have a list of distance restraints that says between which two atoms is the imposed distance between.
And do
And
they cover basically all 5
no,
angstrom local structure?
no, they don't
Okay.
necessarily need to. It's a partial list. It's not for example, oxygens are not detected by NMR, right? So they only,
Okay.
so you get a partial list of whichever molecule, whichever pairwise restraints are observed in the experiment. Somebody assigned this experiment and they deposited it in the NMR DB. So we collect these restraints and we use them as guidance. I mean, we guide AlphaFold to obey these restraints using the laws I mentioned.
Okay. And for the yeah, so I think it's pretty clear how you have this violation loss, right? You basically just use this at inference time to come up with an additional guidance vector field that you add to your diffusion model.
Exactly.
Yes.
Okay.
The only key thing is that you need to do it on an ensemble, because what the measurement that you're observing is an ensemble measurement. So you can't do it per sample because it's not faithful, right? That's not what the experimental measurement is representing.
Okay, so you have like a particle approach. How
Exactly.
many particles do you have usually?
So we used sometimes 32 and sometimes 16. It depends on the observations, but both usually work quite well. And we usually select a subset of the ensemble to get rid of some repetitions of the same conformations in case there are present in more than one. So I think for electron density, we filtered it down to 5 samples. And for NOE, we used 20, right?
We use the full batch.
Oh, we used the full batch. Okay.
Yeah, then let's continue a bit. Later on, I also have some questions about like is
Additionally,
electron density or NOE more helpful, more important, but I'm sure that's better in the results.
So just want to add one more thing here. In addition to ensemble-based likelihoods, you also have your per structure likelihoods. For example, let's say you want to hold some scaffold like that and solve only the problem inside some region. So you could just add that as so as a likelihood term per structure. For example, something like substructure conditioning in chroma.
Yeah.
You could have things like that as well, and we do use it because sometimes we model local electron densities. Then when we hold the rest of the structure in place and only optimize on the block which we're finding heterogeneity on.
Am I right that your MLSP, of course, your MLSP thing is available, and I'm pretty sure that's what I posted as the paper. But your ICML submission, is that on archive or not?
They are both on
Yes.
archive.
Okay.
In MLSB, we use chroma, but it was because it's not sequence conditioned, we lose the whole biology aspect of it.
Also, the way that chroma represents protein without the side chains was a very big hassle because we needed to use a sidechain packer, which introduced a lot of complications and inaccuracies, which is handled in AlphaFold. And also in this paper, we added the NOE guidance, which wasn't present in the chroma paper.
Cool. Okay. Then
Yes.
results.
Yes. All right. So here we can see COVID-19's 8th opening reading frame, which I don't know if you've seen, but it's the best reading frame of COVID-19 spike protein, which is a protein that is responsible for evading the immune system and host pathogen interactions during the infection phase. So the protein exists in 2 mutations, where the mutation is very slight. It's the replacement of 2 amino acids. And we can see the change in structure. It's due to ligand interaction. So these mutations change the way it binds to ligands. But because the sequence is very, very similar, then we get very similar MSA embeddings. So AlphaFold 3 predicts the 2 proteins in the same conformation, but we can clearly see that these two proteins have different conformations in 3 regions. If we use our sampling method using X-ray crystallography, we can see that the guided samples, which are the lighter shades, almost perfectly fit the density, but AlphaFold 3 samples, they stick out of the density completely, and they're also don't have multimodality, so they don't capture both conformations. Another example here is we have a single protein that has multiple conformers of the backbone. So where one of them has an alpha helix and the other does not have that. All chains in the crystal have this property where their backbone is bimodally distributed. Across the unit cell, the proteins exhibit both mods, resulting with the conformations visible in the electron density, so both conformations. We found that about 15% of the PDB exhibit this type of behavior, but because it's the same sequence, AlphaFold and other sequence condition models typically predict just a single mod, because only a single mod is available in the training set, and also because it has the same MSA embedding. If we use our guidance, which is X-ray crystallography, also in this case, we manage to capture both conformations very accurately. If we look at some other proteins that exhibit this bimodal behavior, we can see that non-IID guidance gets multimodality better than its counterparts. So what we mean by non-IID guidance is that we guide over the entire ensemble and not guide each protein separately. So we can see here in this plot that we get biomodality much better than AlphaFold, and also when we do IID guidance.
Yeah, in the case of Noe restraints with NMR structure resolution. So like I explained, the setup is that you're given a list of pairwise restraints, upper bound and lower bound. You're supposed to find an ensemble which obeys these restraints. So, and the way traditionally NMR-based structure resolution is done is by doing an MD simulation. They perform an MD simulation per structure by adding an extra term in the force to the force field, which demands that these restraints are obeyed. And typically, hundreds of such MD simulations are run, and we get the converged structures, and then they pick the top 20 or so based on the energy of the system. So this process takes weeks to run, and maybe even in the optimistic case, it takes a few days. So in this case, for example, on the left is the NMR structure of this PDB-ID, and annotated on that as a darker red shade is the violations. So this NMR structure violates 10% of the restraints that are imposed by the experiment, with an average violation of, or median violation of, 0.32 angstroms. And if you see AlphaFold 3 structures, since they are trained both on crystals, they have very different side chains, even though visibly they look similar. You can on annotated we annotated the violations in dark red shades, and you can see that AlphaFold has, let's say, quarter of the restraints are violated, with, let's say, 2 times more error than what you get from the ground truth. So now we guided the AlphaFold model with our likelihood calculation, and we observe that we get lower, or same in this case, lower number of violations than the PDB structure itself, and about the same error as the median error. And it says that, and moreover, we get this thing in 2 minutes, right? I mean, it's just running AlphaFold with a small additional term to the score. So in some sense, you can accelerate NMR-based structure resolution by replacing MD-based structure resolution to guided AlphaFold. So another thing that we want to mention here is that the typical way of NMR structure resolution is done structure by structure. You take one system, you relax it with MD and asking it to obey some restraints. But this is not true, because the restraints that you get from the experiment are actually imposed on an ensemble. They're not a property of a single structure. So in order to actually do it accurately, there are some crazy efforts like that work on, like let's say, small molecules, which do a batched MD simulation. So which takes a few days, or even in the optimistic case, few days to run, and it produces a more accurate MD simulation to run for NMR structure resolution. But with AlphaFold, we can do the same in a fraction of a cost, and sometimes we get like better results even. So
Okay, so what
yeah.
you're saying here is that we usually get our NMR constraints, and then we somehow initialize our structure. Then we run a bunch of MD, and then we
Yeah.
pick the crystal structure or the structure that fits the NMR structure the best. And here, best is actually just defined as having the lowest number of violations.
Yeah.
Okay.
So this is how it is traditionally done. And this is even doing that is inaccurate because the NMR observation that you have is an ensemble measurement. It's hard to do ensemble MD, right? But there are some efforts from Christoph Lindow Larsen and others. They somehow they actually do a batched MD simulation while asking that the batch of structures obey this NMR restraint. In our case, we do guided AlphaFold and ask the same restraint, and it's faster and many times more accurate and fraction of a cost. So these are a few more examples of different structures that we tried on with this approach. On the 1DEC and 2Li3 are examples where AlphaFold mispredicts the structure completely, or I mean, at least partially. Like one of the terminal regions here in 1DEC and 2Li3 are mispredicted. The fold is wrong. But the experimental measurements suggest a different fold, which is colored in white. The blue are top row blue are AlphaFold's predictions. And you can see that they are also reflected in the number of violations. You see that they are violating around 30% of the constraints that you see there. And just by guiding AlphaFold with the right restraint file, so we get to similar accuracies as we get at least twice as better and like very good alignment with the experimentally MD based resolution of the structure. And on the right, these four examples here, you can often what you see here with AlphaFold is that it predicts very rigid conformations. So you can see that more or less the heterogeneity is lost in between the model, but NMR is firstly done in a solution, and the restraints suggest heterogeneity. And you can see that both in terms of quantitative metrics and even visually, the our ensembles match the MD-based ensembles of NMR much better than
Are
the
these cherry picked or are they completely random?
completely random. We tried it on 10 structures and we just presented those. These are picked based on, we went to some website and downloaded the first 4 structures and ran it. So
okay.
it's not cherry picked. So these were selected with care. Actually, the 1DEC and 2Li3, we actually looked for structures where AlphaFold is doing wrong. So we had this peptide data set of 350 or 400 peptides. We ran AlphaFold on everything, and we picked the bottom 20. And we selected these 2 structures because they have the least corrupt files and when you look at the raw data. So, but this is, in my opinion, it's not cherry picked.
Yeah, it is not.
So, and then we did one more case study. Like one of the popular structures for which NMR experiments is done is a lot of NMR experiments are done is Ubiquitin. So in Ubiquitin, so there is another interesting parameter that you can measure through NMR, which is the coherence of the amide bond. So let's say so this is measured by that. Let's say you have the amide nitrogen-hydrogen bond like that, which is pointed by D. What you can measure through NMR is how coherent is this angle, essentially the direction of the bond. So, and it is measured as a temporal average. Okay, you can measure within a certain time frame. Usually, it's in nanoseconds. How distributed is this quantity? Usually just get the variance. So if you look at the black line, this is an experimental measurement that is collected in the lab, which says that so for example, residue between 7 to 11 is a flexible region because it has a low S2 parameter. It means that it's more dispersed, and it has a larger number between, let's say, 22 to 35, something like that. It has an inflexible helix, and so on. The red line is, sorry, the black line is is the experimentally measured one. And what we did is we ran NOI just by using the NOI constraints and guided AlphaFold. We obtained ensembles, and we measured the coherence of this bond. We can computationally measure this parameter once you have an ensemble. And these are the lines. The red line is AlphaFold. AlphaFold predicts more or less rigid structures for Ubiquitin, and probably because it has seen it a million times in the PDB dataset, always appearing in a particular way. And with guided AlphaFold, without training on this S2 parameter, we are able to recover some correlations between, like, a reasonable correlation with a secondary experiment in NMR. Like, so we guide with one experimental parameter and we check accuracy against the other one. So in this, these scatter plots show you how the experimental S2 parameter is distributed compared to an ensemble, like the computed S2 parameter. For AlphaFold, it's mostly flat, indicating that it always predicts high rigidity. And with IID guidance, you get lesser agreement with the experiment compared to non-IID guidance, for example. It's more accurate to model it as an ensemble. So similar things can be observed when you zoom into one of the, like, for example, this region, it has for K11, which is at 11th residue. And if you look at the amide bond, AlphaFold predicts relatively rigid amide bonds, and the non-rigidity is more more obtained when you look at non-IID guidance compared to IID guidance, even here, for example. So essentially, you're confirming. It seems like the guiding with noise also agrees with other experimental measurements. So
yeah,
that's the paper.
Thank you all for listening. And these are all the amazing collaborators that made this paper come out. Any questions?
So the functions that you use for guidance, they have to be differentiable, right?
Yes.
Mm-hmm.
Then if you have something like the number of violations, you can't directly optimize for that.
Yeah, it's not. We don't measure the number of violation. The violation is measured like that. It's a flat bottom L2 loss, let's say. If you're obeying something, it's flat. If you're not obeying, it's quadratic. Yeah.
The distance to the closest bound. We average it over all the violations.
Then what would you say is, like, right, there are now multiple applications. You can also, I guess you kind of focus on the NMR results now, but you can also do a lot with densities here and just fitting densities. And you're the one where you have 15% of the time the wrong conformation. Did you have a density there, or like the wrong sheet versus helix, or helix versus unordered conformation inside of a crystal structure? Was this improved with NMR data or with density data?
So this was improved using density data. So we see these densities also in the backbone, also in the side chains. These are called usually outlooks, alternative locations for the same node. And it is common, and you do see it in the density, but because this part is flexible, then your density threshold is lower for each of these conformations, because instead of 100% of the time being in one conformation, you have 80% of the time in one and 20% in the other. So usually, if you threshold your density, you wouldn't see the other conformation, as it is more rare. Sometimes it's distributed more evenly, but sometimes you have these types of distributions. But if you guide an ensemble, then you'd get 80% of the structures in the ensemble in one conformations and 20% of the samples of the ensemble in the other conformations.
Would you say these outlooks are usually annotated?
So these outlooks sometimes are modeled. We did notice a lot of cases in the PDB where you can see in the density, and even if you guide, you get multimodality, like clear separation, that it is not modeled. Sometimes it is present in the PDB, but most visualization software only shows the primary outlook, and the primary outlook was used for training the majority of AlphaFold 3. So they were present, the different outlooks, sometimes in training the diffusion model, but the performer, which is the one that derives the contact maps, which are crucial for AlphaFold to work, they were trained using only a single conformation. I mean, they were trained to predict the pairwise distances of a single structure.
Okay. But yeah, with a histogram, you could also supervise it with a distribution, or like with the 50-50 density on both, or whatever density, right? These outlook annotations also contain a density for the outlook. Is that right?
Yes.
Yes.
You have also sometimes a percentage of occupancy.
It is
Yeah.
not very accurate, though.
Yeah, it's not super accurate,
Ah.
but sometimes it's annotated with the occupancy number.
For example, we saw that for places where the density is around 80-20% in the density, the outlook is annotated as 50-50, which is not very accurate. This is, I think, one of the main reasons. Mostly, the predominative outlook is used for visualizations and training.
Yeah. So then we have a question by Noah. Do you have any comments on potential protein structure differences between crystallographic or NMR compared derived, or crystallographic or NMR derived to in situ, e.g. in situ soul, and whether this approach is more or less likely to capture these differences? Is it clear? Should I
So you mean in
explain?
situ? I don't know if you can do in situ crystallography. Because
Sorry, yeah, you cannot do in situ crystallography, of course, but protein structure, as we know, changes in solution and within the cell. And so there are measurements, for example, to show differences between crystallographic and what exists in the cell via FRET and other turf, all sorts of different experimentations. So my question is whether you think this will make you more or less, let's say, dependent on the fact that you have a kind of particular structure out of context, so to speak.
Yeah, I think yeah, we didn't try for a single structure both X-ray and NMR measurements. That would be interesting to actually try. Yeah, this is we did the experiment separately, but actually guiding with both signals at once can get you.
It's a little weird though, because the proteins in X-ray crystallography and NMR, they're in completely different environments. So in crystallography, they are frozen and in a crystal form. So they are not as rigid as in NMR, and usually also the end goal of using NMR or X-ray crystallography is slightly different. For NMR, you are more interested in the dynamics of how the protein changes across time. So the fact that you have a big ensemble is crucial to understand the dynamics of the protein, while in crystallography, you're more interested in determining the 3D structure of a protein that you haven't done before. So there are completely different use cases, but I think it is also an interesting experiment to try to guide with both and see what is happening. But my intuition would say that most of the flexibility from NMR would get lost when we are guiding with the crystallography observations, because they are very rigid. You have one or two conformations that are available, or a few, but they are very, very close to one another, as it is in a crystal, which preserves the shape.
Right, but ultimately we want to know what the protein looks like, where it behaves in the cell, right? That's so yeah.
Yes. So for these types of behaviors, I think NMR is more useful, because you can see the dynamics of how it moves, while determining the initial 3D structure is what is usually done with X-ray. Of course. Any other questions?
Yeah, I mean, Noah.
Yeah, if you don't mind, sure. I don't want to monopolize the conversation, but I'm happy to know also like how this method compares to AlphaFold, because we know, for example, for disordered regions, there's not what are these models doing for these areas, and so I'm curious to know what happens when you guide actually and you have disordered regions in the protein.
Yeah, it's a good question. We haven't tried. I think AlphaFold usually predicts very structured proteins. It can't predict IDP side from what I, let's say, roughly know. But it could be interesting to see if by guidance, you can impose the fact that it's an IDP.
Mhm.
Right? It's your experiment is suggesting that it's an IDP, right? But
Right.
AlphaFold is saying it's rigid.
Yes.
Could be interesting to see if you can enforce it to.
Absolutely.
There's also a good reason that AlphaFold fails in many of these cases if you think of the way AlphaFold was trained. So it was trained using almost exclusively crystallographic data. So they have very specific and rigid behavior. So even just if you want to use AlphaFold for NMR, you are trying to get it to a different domain. So this is one of the reasons we saw that for NMR observations, if we compare the order parameter, the S squared of other NMR observations and what AlphaFold gives, it always gives a rigid structure because this is the type of structure it was trained on. So this is one of the reasons I think it is not suitable for IDP. Also, I don't think X-ray crystallography is used for IDPs.
No, you can't see anything.
So,
You
yeah,
don't see anything.
because
You can't crystallize.
yeah, so
Right.
this prior is a bit ill-suited for this purpose, but it could be interesting to try to guide maybe using NMR observations on IDPs. Could be interesting.
Very cool. And that actually leads to the last question, which was, could you expand this to account for like predicting PPIs or kind of multimers in the same ways that AlphaFold tried to do it?
Yeah, I think for sure, because AlphaFold handles complexes, right? It's also protein ligand complexes, protein complexes, and it should natively, I think, extend, because there's no complication that we can think of except technical ones. Yeah.
Yes.
Very cool. Thank you, guys. Very interesting.
Yeah, then also from my side, a big thanks. And I think we can comfortably call it a day here. And yeah, it was very interesting to me. And this, I guess, ensemble guidance is kind of a bit of a, I mean, we have things like particle guidance, right, where we try to enforce more diversity in our diffusion model. But here now we kind of have a very nice guidance term that is applied to the ensemble that we need to, or that we can follow. Yeah, pretty cool. Any last words from your side, or should we call it a day?
Oh,
Thank you for having us.
All right, sounds good. Then enjoy Austria
Yeah.
and Vienna, and see you around, and see you around everyone else. Bye.
Thank you.
Thank you. Bye-bye.